Hi everyone, my name is Melissa Hernandez and I'm doing the Evolution in the News project for this week. The article I'm using is called Biochemists Discover Mechanism That Helps Flu Viruses Evolve. To start off, I'll give a brief summary of the research from both the general audience article and the peer-reviewed article. We noticed that the MIT team experimented with the flu virus and how the chaperone proteins within the virus hijacked the host cell. And this leads to the rapid evolution of the flu virus, which leads to us, the general public, get the flu vaccine every year because it's constantly being updated. The MIT team particularly experimented with the relationship between the chaperone proteins and the virus itself, and we noticed that the viral envelope is the primary point of contact. The summary of the article also states that there's future work with protein folding and there are targeted enzymes as well to see if it can delay evolution of the flu virus or even affecting the rate of the viral evolution overall. To continue on to the key findings, the hypothesis of the MIT team during this study was whether or not chaperone protein levels will lead to faster viral evolution or if they can be inhibited enough to slow it down. So the importance of this study is that the flu virus in particular is constantly mutating. This is why we need to have the vaccine updated every flu season in order to keep up because it keeps changing every year. The main goal of this research was to take the findings and see if we can kill the virus in the host cell before the virus decides to become drug resistant, and this can lead to vaccines becoming less frequent for us. The methods used, we see that there is a hemagglutinin protein, which is a gene of interest inside the virus, and we notice that, again, the primary point of contact is the viral envelope, and this is how we have the interaction with the host cell. And this primary point of contact is the main reason for the rapid evolution. So the experiment set overall was divided into two sets of cells. The first set was low protein folding activity, and the research team went in and inhibited the specific HSP90 chaperone protein, which is the hemagglutinin protein. Then they took the second set of cells, which is the high protein folding activity set, and this is where they enhance chaperone protein levels and they also enhance the interaction with the host cell, so more of a contact between the host and the virus on the viral envelope layer. And they also added a heat shock response in there as well to see if that would change anything with the chaperone protein levels. They were both infected with the same flu strains at the same time, and they analyzed the results and noticed that the one with the high protein folding activity did actually enhance viral evolution by 200 generations more. So we see that if the chaperone protein HSP90 can be inhibited with all viruses, then there could be a stop in the viral evolution overall, or at least slow it down. For media and research, I think the hook of the article was important because it discussed the people first, how the general public is affected by this flu virus study and what could come out of it for them. So I think it was important to note that the reason we have the flu vaccine every year is because of the constant mutations of this virus. So discussing the fact that we could change this by just having this research, then people were automatically hooked to continue and read the article. So there was also a discussion on the drug mechanism and how this could lead to further investigation into the flu virus and how drugs would affect it later on with the resistance of the virus. We also had comments from the lead researchers of the MIT team, which was a good hook for the article as well. And the overall flow of the research article was great because it went in chronological order. For the controversy, I think it's important to note we don't know if the general public believes in viral evolution. There might be a lack of understanding with the genetic mechanisms of the virus, so it's possible that many people may not believe that a virus can evolve. This would be the one controversy for this article. The article's strong points were, like I said, the chronological order. They gave a great genetic backgrounds of the virus with enough detail. And at the end, they inputted future target phenomenons, which was important to let the public know that there's more research going on with this specific virus and the specific experiment itself with other viruses. The scientists addressed were the main ones, Shoulders and Lindquist, and they gave plenty of comments about the research in there as well to state their point of view and the reasons behind the research for them. In conclusion, and to give a discussion for the Evolution in the News class members for this week, overall, the MIT team was obviously successful in investigating the flu virus and how chaperone proteins affect viral evolution overall. We noticed that the high protein folding activity was the more active one in producing further generations of the virus, and if we can potentially inhibit this in all viruses, then we can delay the viral evolution overall. There is possibility of future studies with other quickly mutating viruses, and this includes HIV. So if there can be an experiment with the protein folding or the enzymes or the viral envelope of HIV in contact with the host cell, it is a possibility. And for the class discussion, I want to address a question of whether or not you think this study should be continued or reviewed. Do you think that the current flu virus method is enough for the general public to feel at ease and fully protected from the flu virus? So if there was more research into this, is that important enough for the general public to feel better about not getting the flu versus having to get a flu shot every single year continuously in order to keep up to date with the mutating virus? If you would like to access the articles to get more information, here are the references. Thank you, everyone.